Welcome everyone to this week's edition of Extracellular Vesicle Club. EV Club was founded by Ken Whitwer in 2020, but has now grown into an event of the International Society for Extracellular Vesicles, that is ISEV. My name is Metka Lenassi, and as is probably true for everyone joining us today, I'm an enthusiastic EV researcher, but I'm also working with Ken on the Science and Meetings Committee of the ISEV. And I will be your host for today. And now it is my great pleasure to welcome Ahmed Ibrahim and Alessandra Ciullo from Cedar Sinai to present their exciting work on cardiosphere cell-derived EVs, which have a great promise as cell-free therapeutics in heart diseases. And to better understand biodistribution of these EVs and of their cargo, Ahmed and Alessandra developed a method called detection of unmodified EVs using RNA tracer. And I'm very interested to learn more about that. So just a short note, I encourage everyone to place any comments or questions in the chat box during the talk, and it will be possible to unmute yourselves at the end and ask these questions in person. So that's it from my side, and over to you, Ahmed. Please start sharing your screen.
Thank you so much, Metka, for this lovely introduction. And I want to thank the organizers for giving us this lovely opportunity to be able to present our work. This is the first sort of social event that we have since the pandemic, so we're hungry for your questions and looking forward to sharing our story, and we're all very excited. Today's talk will focus on, as Metka mentioned, the paper that we published recently, and it's titled Biodistribution of Unmodified Cardiosphere-Derived Cell EVs using single RNA tracing. And I wanted to give everyone just a very quick rundown of the topics that we plan to discuss. I wanted to discuss the value of tracking EVs in biological samples. And then I wanted to focus specifically on CDC EVs, as these are the therapeutic agents that we've been working with for some time now. And then an introduction to our method and how it was that we were able to focus on one RNA cargo as a tracer for EV biodistribution. And then I'll hand it over to Ali, who'll talk about in vivo findings that we obtained using that method. And then finally, she'll talk about some of the in vitro findings as a finer dissection of studying EV kinetics in situ. Then I'll hand it back to me to talk about broader implications, and then discuss some of the study limitations in future work. Most of the discussion today will focus on EVs from a therapeutic perspective. We know that historically EVs have been investigated as prognostic and diagnostic markers of disease, but in our corner of EV biology, we focus primarily on EV translation. And as you can probably imagine, being able to track EVs in vivo in a biological system is important to establish the pharmacokinetic profile. That includes, the absorption, the distribution, the metabolism, and the rate of clearance. But it's also important understanding pharmacodynamics, because if we can understand the target tissue that these EVs affect, we can begin to understand their effect on the disease specifically. So if they're uptaken by macrophages, it's likely immunomodulatory. If they're uptaken by fibroblasts, fibrosis might be involved. And if they're uptaken by, in our case, cardiomyocytes, they might be cardioprotective and so on. So that's important in that regard. Now in vitro, understanding EV or measuring EV uptake is important to measure kinetics. It's also important to measure to understand their fate once they're uptaken by cells. There's been a long discussion about whether EV tropism is real and how specific that is. And also, as I've mentioned previously, understanding cargo fate. So are these EVs being taken to the lysosomal pathway and being degraded, or are they being taken to the ER for retrograde sorting and then being used as functional signals? So a little bit of a primer on CDC EVs. So I worked on CDC EVs as a graduate student back in 2010, back when dinosaurs roamed the earth. And cardiosphere-derived cells are these very therapeutic population of cardiac-derived stromal cells that have pluripotent capacity. And they've been shown in both preclinical and clinical settings to repair tissue after injury. And if everyone can see my cursor, the clinical trials on the right reflect studies that were done using CDCs, Caduceus being the first of them. This was a phase 1 clinical trial in myocardial infarction, followed by heart failure and Duchenne muscular dystrophy, pulmonary hypertension and heart failure with preserved ejection fraction. So they've shown disease-modifying bioactivity in all of these disease indications. And the 4 primary mechanisms of action, or the functions, have included tissue growth and survival, immunomodulation, likely through the innate immune response, the stimulation of angiogenesis, and scar clearance. And that's going to be important because what we found out later on was that these effects, as many, by the way, people in the cell therapy field have found, that the primary function of these cells is indirectly through the secretion of these extracellular vesicles, which we know are multi-classed and contain a plethora of biological signals. And so we've shifted our focus from the cells to the EVs, not only for mechanistic reasons, but also for practical reasons, because they're a lot wieldier, they're unchanging because they're non-living entities, and they're just easier to translate to the clinic. Since our discovery back in 2014, this field in our lab has blossomed into maybe 3 main areas of investigation. One that looks at the mechanistic aspect of EV signaling, which I think is very important as you begin to understand how EVs function in vivo and in vitro. We've also expanded to investigate other disease models, simply because EVs affect the inflammatory responses that are germane to a lot of disease processes. And then finally, being able to understand the mechanism, we've been able to engineer them based on the mechanistic findings. As this is an ISEV event, we have to discuss the ISEV guidelines, which are very important. The EVs that we'll talk about today were isolated using a 15-day serum starvation method, using IMDM as the sole ingredient in the conditioned media. The primary isolation method of the EVs that you'll see today has been by ultrafiltration by centrifugation using 100 kilodalton cutoff, but where confirmatory studies are important, we also use size exclusion chromatography as sort of the gold standard of investigating EVs in the absence of any of the soluble proteins that might be co-precipitated with the previous isolation method. As far as doses go, in vivo models, we use 2 × 10⁹ particles in mice. And in vitro, we use a proportion of 100 EVs per cell. And as you can see below, this is a nanosite plot showing the average size of these EVs to be around 150. These are the conserved EV markers, as shown here by TSG101, some of the tetraspanin, CD81 and 63, HOC protein 90, and of course the absence of the contaminating cell marker calnexin. Off to the right, you see electron micrograph images of EVs, and of course, as we all appreciate, EVs come in multiple sizes, and they come from 2 primary origins. So one of the limitations that I'll discuss later on is that the biodistribution that we study is of the entire EV population. So there might be more than one subtype. Even though we call them small EVs in the literature, I think the field is still trying to understand what delineates one population from the other. But that being said, our first area of investigation was, like many groups, on microRNAs and how we can use microRNAs to track EVs. The problem with microRNAs is that they're incredibly evolutionary conserved, so there's no real way to measure the uptake of EVs by one enriched microRNA or another, simply because what you see in humans will also be present in models such as mouse and rat. But what we also found, which was very exciting, is that CDC EVs have a very unique signature and a higher diversity of different RNA classes. In fact, if you look at this blue slice, microRNAs comprise 7% of the total RNA content of EVs. And a much bigger proportion is this slice of yRNAs. And yRNAs are RNAs that are known to participate in ensuring DNA stability during replication. Not much else is known, although this is a very active area of research in the lab, compared to fibroblast EVs, which are mostly tRNAs. And while tRNAs have been sort of dismissed as junk RNA, more and more research is showing that they're involved in other active signaling processes in their own right. Okay, so NT4 is a fragment of this larger human yRNA4 gene, and human yRNA4 is, as the name implies, is human specific. So this is absent in mice. And the interesting thing is the single most abundant RNA species in CDC EVs by a long shot was this 24 nucleotide fragment in these EVs. And as you can see in panel C, there's this species specificity is reflected in the absence in mouse dermal fibroblasts in human, and the enrichment in human dermal fibroblasts. We also show in these plots collectively, in these figures collectively, that they're exclusive to EVs, so they're inside the EVs and not secreted on their own. This is shown by a 10K preparation detected in similar amounts in a 1000K preparation. In panel E, you see an RNase protection assay. So, they are encapsulated inside CDC EVs, and that we're able to measure them with some certainty. And whether it's by 100K isolation or size exclusion chromatography, they're present. Now, IMEX is an immortalized CDC EV product that is a product of, again, mechanistic investigation that we were then able to use to engineer the cells. And they too are enriched in this NT4. What we show here is that the assay that we developed, the qPCR assay, to be able to detect NT4 specifically is efficient, as shown by this very linear plot of increasing amounts of CDC EVs in, I believe that's liver tissue that we're working with. It's also specific, so these are melting curves of different concentrations, again, of EV, so that it shows that across different concentrations, you're able to very specifically detect that signal and not some other background amplification. And then here we also show that they're actually this assay is quite sensitive. So the barrier of detection is fairly high resolution, so 400 copies per milligram of tissue. So if you have 400 copies of this NT4, whether by the RNA itself or by CDC EVs, we will be able to detect a difference compared to the background of an otherwise naive tissue sample. And I will now turn this over to my wonderful colleague to tell you about the exciting in vivo data that comes from this method.
Going more inside into the body of the work, just to recap, so having identified an AB-associated tracer that is NT4 and validated, as Ahmed just shown, a reliable strategy for tracking a modified AB and their cargo, we sought to understand how CDC AB biodistribute under healthy and disease conditions in vivo, but also in vitro. So starting with the in vivo data, first of all, for translational relevance, we used intravenous delivery as route of administration of choice for our CDC-EB. And based on preliminary data, we determined that 1 hour is the optimal time point for the highest sensitivity for our method. So to determine the biodistribution of CDC-EB in healthy animals, 10 to 12-week-old female mice were injected intravenously via the femoral vein with 100 microliters of serum-free media containing, as we just mentioned, 2 times 10 to the 9 extracellular vesicles, or just serum-free media only for the vehicle group. And 1 hour after administration of CDC-EB, we collected, so mice were sacrificed, and the main organs of IB retention, as known in literature, that were heart, liver, kidney, spleen, brain, and lungs, were collected. We also decided to isolate blood and urine to get some ideas of clearance, because if we can detect, for example, EB in the urine by 1 hour, this will give us a bit of a hint of how these EB are cleared. So from all these organs and body fluids, we isolated RNA for quantification of NT4 copies using our qPCR-based method to track the biodistribution. So as previously shown by Ahmed, we have a standard curve using animal tissues where we add the different concentration of our CDC-EB, and this way we calculated the number of EB present per milligram of tissue, and also per, and of course, per microliter of plasma and of urine. So I want to take your attention, first of all, before discussing the real data about these letters and symbols. So for each organ, we performed one-way ANOVA, And this is the significance to every other organ. So for example, here you can see that the heart is significant in the heart, the retention, so the uptake is significantly higher than lungs, brain, spleen, and kidney, and so forth for all the data. So what we found that is very interesting is that from all the organ on a per milligram basis, the heart retained, so cardiac tissue retained more extracellular vesicles than the other organ. And this was followed by the liver and then spleen and kidney. By organ, so this graph, the graph C, is a little bit different because of course liver is a larger organ, so it retained a higher overall numbers of EV, and this is in agreement with previous reports in literature. But in contrast with previous findings, however, the second most retaining organ on a per organ basis is the heart, followed by the kidneys. So as I said, the retention by the liver and the kidney is supported by several reports in the literature using various and different approaches. However, the higher retention that we find in the cardiac tissue is a novel finding and may suggest, for example, a preferential uptake by certain cells. Furthermore, at 1 hour post-administration, a significant portion of extracellular vesicles remained in circulation. So this is a little bit in contrast with previous findings demonstrating that in mice, demonstrating that EV were cleared very fast from the circulation. We want to mention that, you know, different methods they were used, and there are pros and cons, of course. But we can also add that there are also recent investigations showing a higher retention in the plasma with other methods like luciferase exosomes. We were also able to detect some traces of extracellular vesicles in the urine, indicating rapid clearance. So to conclude this slide, 1 hour post-administration, the majority of EV cargo is found in plasma, followed by the liver and then the heart and kidneys. So then we wanted to ask how the biodistribution changes when CDC-EB are administered through an alternate method. So for this reason, to evaluate whether the site of intravenous administration affects the biodistribution, we used retroorbital administration as a comparator through the femoral, through the injection through the femoral vein. In this case, so for the retroorbital administration, CDC-EB, they are not directly injected into the vein, but they go into the vein through the sinus. So also in this case, we injected 2 × 10⁹ extracellular vesicles, and then after 1 hour, as I previously mentioned, we collected the main organ and also the body fluids, and we performed qPCR to detect NT4 copies into the tissues. So also in this case, per milligram basis, heart was the organ with a higher retention, followed by liver and kidneys. So on a per organ basis, like the femoral injection, the liver was the organ where the retention was higher, followed by heart and kidney. When we compare here in the graph D, the 2 routes of administration, so the femoral vein injection through and the retroorbital administration, we can see significantly higher levels of CDC-EB with this method into the brain and lower levels into the lungs. And also, similar to the femoral vein injection, the majority of administered CDC-EB remained into the circulation at 1 hour post-administration, and also some traces were detected into the urine. So taken together, what we can conclude is that the retroorbital administration of CDC-EB results in similar biodistribution, except for elevated retention in the brain tissue and lower retention into the lungs. Then another question was the fact that, we wanted to determine whether the engineering of the producer cells, in this case, immortalization, as Ahmed described at the beginning, so in the introductory slides, affected the biodistribution of the extracellular vesicles. So for this reason, we evaluated the biodistribution of EB produced from an immortalized CDC cell line. The name are IMEX of these exosomes. So this immortalized CDC we developed back in 2019, where we genetically engineered CDC to retain their potency, but at the same time to be immortalized. So also in this case, we injected IMEX into the femoral vein, same concentration, same time points. We collected the main organs, body fluids, and then qPCR, and we evaluated both per milligram retention and per organ retention. So similar to CDC-EB, the liver and the heart were the primary location for EB biodistribution, followed by the kidney. So we can conclude that so extracellular vesicles from immortalized CDC had a similar biodistribution profile compared to those derived from primary CDC, so that immortalization step doesn't change the biodistribution, but retain the same biodistribution characteristics. Next question was, because CDC-EB are used primarily for heart disease indication, and of course we are a cardiac lab, we wanted to compare CDC-EB biodistribution in healthy animals and in animals after an injury. So in this case, cardiac ischemia reperfusion injury. So in this case, about this protocol, animals underwent transient coronary ligation to induce IR. So essentially ischemia when it was induced in mice. Then after 45 minutes, we allowed the animals for reperfusion, and we injected through the femoral vein as our protocol, CDC-EB 20 minutes after the reperfusion. So also in this case, the major organs were collected and also the body fluids. So, and we also evaluated the retention and biodistribution profile per milligram and per organ. So as observed also in the healthy model, the two top contenders of EB retention were the heart and the liver, both on a per milligram and then per organ, first the liver and then heart, followed by the kidneys. And, but also in this case, so as previous administration routes and the previous results in healthy animals, the majority of extracellular vesicles were retained into the circulation, and some traces also detected in the urine indicating rapid clearance. So then going more inside, if we want to compare the biodistribution in healthy and diseased animals, we actually see a fair amount of differences, and we can make some considerations. So first of all, we can say that using the ischemia reperfusion model, we were able to account for around 90% of the injected extracellular vesicles, so a number significantly higher than into the healthy protocol. The second thing that we can add is that animals with ischemia reperfusion injury retained higher levels of CDC-EB or CDC-EB cargo, most of all, into the heart, into the liver, and into the brain compared to the healthy protocols, with similar trends observed also into the other organs, though they did not reach the level of statistical significance. So we were not surprised to see higher retention into the injured heart, but for example, we were surprised to see higher retention, for example, into the liver, perhaps because of secondary inflammatory consequences. Most surprisingly was the brain, but these results needs need further investigation. So these findings, most of all, support early observation showing that the modification of the surface of injured cells can facilitate or can change AV trafficking and uptake, especially in the injured muscle. We were then curious about, which portion of the heart had the highest uptake, the highest retention in terms of EV cargo. So to determine whether the site of injury retained, in particular, the higher amount of extracellular vesicles, we obtained tissue from the right ventricle, the left ventricle, and the infarcted region. So just a small highlight about the protocol. So in the ischemia reperfusion model of cardiac injury, the left anterior descending coronary artery, which perfuse around 50 to 60% of the left ventricle, is temporarily occluded by the protocol, followed by reperfusion. So this leads to ischemic injury and damage to the left ventricle with no direct insult to the right ventricle. So in congruence with previous findings, we see that AV were retained in the infarct area more to the uninfarcted area, so of the left ventricle, and more compared to the right ventricle. Now that we have shown that extracellular vesicles are retained by heart and liver, until now we have shown in a healthy and diseased model that the majority of extracellular vesicle cargo in terms of NT4, as we do in our protocol, were retained more into the liver, into the heart, in healthy animals, and then in the injured condition, we have an overall higher uptake and retention in all the organs, but specifically again in the heart and in the liver. And in particular, when we analyze, the injured tissue, it is specifically into the injured area compared to the healthy area of the heart. We then ask the question of how accurate and how reflective is this approach if we perturb the uptake mechanism of extracellular vesicles in vivo. So to dissect the mechanism of extracellular vesicle uptake in tissue, we decided to use two inhibitor of uptake, of different kinds of uptake. So in particular, we used heparin, as involved in extracellular vesicle surface docking, and we use dinosaur hydrate that is involved in clathrin-mediated endocytosis and micropinocytosis, respectively. So we injected intraperitonially both heparin and dinosaur hydrate 10 minutes before, so after ischemia reperfusion model. So also in this case, we have injured animals with ischemia reperfusion. 10 minutes after reperfusion, we injected intraperitonially heparin and dinosaur hydrate, and 10 minutes after, we injected exosomes, so same administration route and same concentration. As you can see here, intraperitoneal administration of heparin 10 minutes before extracellular vesicle administration led to a significant reduction in uptake in several organs. This includes the liver, the spleen, the brain, lungs, and surprisingly, it also decreased the retention of extracellular vesicles in circulation. That can be related to heparin-mediated mobilization of the macrophage in this model. So unexpectedly and surprisingly, the retention by the cardiac tissue was unaltered, which may suggest and points to different mechanisms by which cardiac tissue or injured tissue in general binds extracellular vesicles for the uptake. When we evaluate the biodistribution profile of animals treated with dinosaur, however, we have a dramatic reduction of AV retention in all the tissues of about 50%, even if more in some organs. So this essentially demonstrates that the retention of extracellular vesicles in tissue is not driven by accumulation in capillary beds, but rather on clutter-independent endocytosis processes or macropinocytosis of the extracellular vesicles particles. Surprisingly, also in this model, in this dinosaur hydrate administration model, we observed significantly lower retention of extracellular vesicles in plasma. Moreover, to investigate whether the inhibition of the uptake led to the abrogation of the therapeutic effect of CDC-AV, because I want you to remind that cardiosphere-derived cells, extracellular vesicles, are used for cardiac disease indications, and we show that they exert therapeutic effect. So we decided to induce again the model of ischemia reperfusion, and then to inject CDC-EV alone that we know that exert therapeutic effect via the femoral vein, with or without pretreatment with the two inhibitors of the uptake, so heparin and dinosaur hydrate. We collected the hearts after 48 hours, and we measured the infarct mass, mentioned here as TTC, that it is the staining that we use. And as you can see here, like the images. As you can see, animals infused with CDC extracellular vesicles only had reduced infarct size as expected, compared to controls, while animals receiving CDC-AV with heparin and dinosaur both had the scar size similar to vehicle controls. So, for example, it's interesting to note that despite the retention of the uptake capacity by the heart in the heparin treatment, the CDC-EV effect was attenuated. And this can be explained by the fact that, we previously shown in our work that, for example, macrophages play an integral role in the mechanism of action of cardiosphere-derived cells and extracellular vesicles, of course, in cardioprotection. So therefore, the inhibition of EV docking or endocytosis, we can conclude that block cardioprotective effect of CDC extracellular vesicles. Finally, for most of this talk, we talked about, the in vivo model, but we wanted to go over, go down with a higher resolution and more reductionist model, that is the in vitro model. So to more finally dissect the uptake kinetics of individual cell types that make up the cardiac microenvironment, we decided to isolate primary cells from rats in this case. So we have neonatal rat ventricular myocytes, so essentially cardiomyocytes, cardiovascular, microvascular endothelial cells, cardiac fibroblasts, and bone marrow derived macrophages. And we decided to administer 100 exosomes, 100 extracellular vesicles, per cell, and we evaluate the uptake at different time points, in particular 20 minutes, 1 hour, 2 hours, and 4 hours after exposure to CDC-EV. We observed, as you can see in the panel B, variable kinetics of CDC-EV uptake in between the cells tested. So expectedly, macrophages, that are the blue line, had the fastest and the highest overall uptake, followed by cardiac fibroblasts in orange and endothelial cells. And on the other side, here in purple, cardiomyocytes had the slowest and the lowest uptake, overall uptake of extracellular vesicles. I want also to add something more. So as you can see here, for macrophages and cardiac fibroblasts, the signal was retained over the time of the 4 hours where we measure the uptake. It is not the same, for example, for endothelial cells, where they start to uptake after 20 minutes, and then there's a peak at 1 hour, and then the signal was lost after the 1 hour until the 4 hours, and it is maybe due of internal degradation of the EV cargo, that is what we measure, or a re-release into the media of EV and EV cargo. So these results suggest that macrophages, cardiac fibroblasts, and endothelial cells are the effective recipients of CDC extracellular vesicles, and that the effect of CDC-EV on cardiomyocytes is likely nominal. We then wanted also to demonstrate for in vitro experiment that our method was able to detect a decrease in uptake, as we show for the in vivo model. So in this case, we used cardiac fibroblasts just to remove the confining effect of phagocytosis that we may have using macrophages. And as you can see here, like the in vivo model, we use the two inhibitors that we also use in vivo, dinosaur, a different concentration, and we show that we can see compared to extracellular vesicles alone, we can see a decrease in the uptake of the cells, and we also use heparin at a different concentration. Finally, using this technique in vitro, we demonstrated that essentially macrophages, cardiac fibroblasts, and endothelial cells are the principal recipients of extracellular vesicles compared to cardiomyocytes, and then thus cardioprotection is likely mediated by those cell types rather than a direct influence on the myocytes themselves. So I will turn over to Ahmed for the conclusion and the final considerations.
Thank you so much, Ali. So we've shown a lot of data today, but I wanted to focus sort of on the take-home message of this paper, which is, as Ali so eloquently described, this is a method for tracking native EVs and EV cargo in cells and tissue, and that's important because by and large, most of the approaches used to track EVs have involved some form of manipulation of EVs, whether it's transfection with some sort of foreign RNA or some sort of imaging agent. But this is the first, well, among the first that we know of that tracks native unmodified EVs, both in vivo and in vitro. And the main findings are the following, that most of the uptake that we observed on a per milligram basis in the heart, followed by the liver, and then so on. In terms of the total amount of EVs that are retained, the liver obviously retains more simply because it's a larger organ. The other important message to consider, which is very much in line with a growing body of evidence regarding the behavior of EVs in injured tissue, is that in an injured environment, the retention is much higher if the signature remains the same. So the heart remains the principal uptake organ on per milligram basis, followed by the liver. Now, there's a caveat to that I'll mention later on, but just to round this off, when we dissect sort of the microenvironment, the main constituents, and I said before, this method is very important in looking at the pharmacodynamics, because based on the principal uptake cell type, gives you a hint as to what the mechanism of action might be. And we identified macrophages and cardiac fibroblasts. If you recall earlier in the presentation, the two top sort of mechanisms of action of CDC-EVs is antifibrosis and immunomodulation. So this very much falls in line with the observations that we're making, and it further establishes the mechanism of action of EVs. We also showed that there's this very important corner of EV biology, and there's a fair criticism that the EVs that we're injecting, or the EVs that the effects that EVs produce in vivo across different bodies of evidence were elicited by some non-specific transient inflammatory response. And here we show that if you block uptake specifically of the EVs, then you lose that protective effect, which lends more credence to the idea that the EVs themselves are very intentionally bioactive disease-modifying agents. Now, one of the main sort of critiques of this work raised by some of the reviewers were that, yes, this is, specifically CDC-EVs, but is this more broadly applicable? And the answer is yes. While NT4 is really specifically CDC-EVs, the idea was to propose or to show that any RNA cargo with a specific set of parameters can be used to track EVs, and those parameters include sufficient abundance in CDC EV in the EV population of choice. The model system that you're investigating has to have a low enough background, has to be exclusive to the EVs compared to the model itself, and you have to be able to optimize a qPCR protocol that's specific enough to be able to pick up this RNA in tissue with sufficient specificity and sensitivity. The other major contribution is that it enables, obviously, the pharmacokinetic and also pharmacodynamic properties of EVs. It also provides further mechanistic evidence to the signaling role of EVs as we understand them, meaning that they have to be uptaken and they have to be processed in order for an effect to be seen. And that provides a really nuanced understanding of uptake by different tissues. Now, as all studies there are limitations. For instance, this approach is limited to, by and large, is limited to a terminal setting, because aside from urine and plasma, you'd have to euthanize the animal to get the tissue, as opposed to something that's maybe more visual, that you can track sort of in vivo and in real time. The other challenge is that it does not delineate between the EVs and the EV cargo, because really what you're measuring is the presence of the RNA cargo. There's no guarantee that this RNA cargo is retained inside the EV. You could be measuring the RNA cargo long after it's been unpackaged and inside tissue. So there's no delineation between those two contexts. The other study limitation is that we looked at EVs wholesale, meaning that we didn't parse out the microvesicles from the small EVs, from the exosomes. Now all the new classes that have been identified, the exomeres and so on. So this was really looking at EVs collectively as isolated by size exclusion, but primarily by 100 kilodalton isolation method. The other caveat that I mentioned earlier when I was showing you this graphical abstract is we need to be cautious about the way that we conclude where these EVs go, because remember, CDC EVs are cardiac derived. So the idea that they're retained primarily by the heart on a per milligram basis may be a product of where these EVs come from. It could be a tropic phenomenon that we're looking at that may not translate well, or that may not be generalizable to EVs from fibroblasts or from hepatocytes or from macrophages. And so it would have been helpful to also compare an EV source from other tissue to see if the heart generally just takes up more EVs in general, or is it latching on to EVs from a similar tissue type. And then finally, there are some organs that we didn't look at. Obviously, we looked at most of the organs that one considers when you think about EV uptake, but we left out important tissue types like skeletal muscle. That's important because we investigated Duchenne muscular dystrophy as part of our research program, but also other organs like the skin, which is really the largest organ in the body, and one that would be, we would expect to see EV uptake there as well. So these are some of the limitations. The future studies obviously would be to look at the biodistribution of specific EV subclasses. And there are now instruments that really enable us to do that, including asymmetric flow field flow fractionation. So we can isolate EV populations based on a very narrow window of size. And if we can find out what the abundance of something like NT4 is, adjust for that value, and then inject that specific population, and then see if the heart, for instance, uptakes small EVs more readily than it does microvesicles, or the other way around. The other future step that can be taken is to selectively knock down different uptake receptors to see which ones, let's say, are more consequential than others. We can also analyze the biodistribution and retention at different time points, because you can see most of the data that we collected, actually all the data that we collected, was at 1 hour post, and that's not enough sufficient to measure half-life. You want to be able to measure it at multiple time points. Then finally, our work stops at uptake, but what happens after uptake is equally important. Where do they go? Do they go to the ER, or do they go to the lysosome, or are they spat back out? The data that you saw with endothelium, with endothelial cells, it suggests either, as Ali mentioned, immediate degradation or immediate expulsion. And those two things are important, again, when considering the fate of these EVs in vivo. Well, finally, I want to thank our wonderful team who without which this work would not be possible, starting with Alessandra. Alessandra joined us around the time of the pandemic, and her tireless efforts and her sort of brilliant way of designing experiments was really key to the success of this project. So a lot of the credit goes to her. Of course, we have the rest of the team: Cheng, my research associate; Liang, our capable animal surgeon; Candice, our surgeon resident who joined us for 2 years and made some novel findings of her own; Kyle, who just graduated a couple of weeks ago, defended his PhD quite handily, so congrats to him; and of course my mentor and my enduring friend Eduardo, and all the members of our collective lab. And thank you, Metka, for having us, and thank you, Ken, for your infectious enthusiasm, and we're happy to take questions. Thank you.
Okay, thank you a lot, Ahmed and Alessandra, for this really clear and interesting presentation. And there's already some questions, so I'm gonna now allow all of you to unmute yourselves, and I think that we can just try to just invite all of the participants who have questions to pose them in person and maybe follow up their discussion. So I would like to invite Noel Miller to ask his question.
Hello, I was just wondering, when you're talking about, CDC-Ds in the clinical trials, I was wondering if any of them were in phase 3 yet, or if all of them were listed like in that phase 1 and 2 stage. Is that what you're mentioning?
Thank you so much for this question. I'll take it. So the clinical trials that I showed, Noel, they were all in the cells themselves, cell therapy. And they were phase 1 or phase 1/2 that I listed. So I believe Hope is in phase 2. Phase 2 at the moment. Just published. Right. So CDC-EVs have yet to be applied in the clinical context.
F. Messaggio, so probably this is not the name, but maybe if you.
Hi, can you hear me?
Hi. Yes, go ahead.
Yeah, my question was like, when you show the Western blot at the beginning, I was curious if you tested the CDC cells and if they were positive for any EV marker.
Yeah, so thank you so much for the question. Yeah, we also tested CDC. As we show, we didn't show really the same markers, but yeah, they are also positive for CD63, that all the tetraspanin CD9, CD81, HSP, 70, 90. What we showed there with the cells and the exosomes, of course, was the absence in the extracellular vesicles or of cellular contaminants like calnexin, for example.
And then Greg Bruman.
Hi, yeah, I was just curious on how the dosing strategy was determined. I noticed that there's it was a kind of a per cell basis for in vitro, and then just a bulk treatment for in vivo. I was just wondering how you determine those concentrations.
Thanks for the question, Greg. It's a really thoughtful question. So really based on experience. So when we first started working with EVs, we used all sorts of doses ranging from 0.1 EVs per cell to up to 10,000 EVs per cell. And what we found is that anywhere between 100 EVs per cell to 1,000 EVs per cell, depending on the cell type, is where we saw a biological effect. Now, as far as the dose in animals is concerned, we did something similar. The very first studies were done using polyethylene glycol precipitation. That's during the Stone Age of EV investigations. But we later found out that we injected e to the 8th, e to the 9th, e to the 10th, and we found that between e to the 9th and the 10th there wasn't much of a difference. And so we stuck to e to the 9th particles in mice, and I believe it's the 10th in rats. The interesting point, and that's one that you touch on a little bit, is how to adjust the dose between different sizes of animals. And what we found is it's not necessarily linear to the size, but rather to the number of cells that we have typically injected in the past, that proportion anyway. So I hope this answers your question.
I think he's happy. No, he doesn't have any further comments. So.
Good. All right.
So there, Philip Askenze has, I think, 2 questions at least. So maybe just please unmute yourself. And
yes.
Okay, go ahead.
So this is very important work. So for calculations, did they get an idea of So I have a couple of questions. The first is the question of the copies of EV per copies of what you're following per EV. What did that turn out?
Yes, thank you for the question. So for the primary CDC EVs that we looked at, I believe it was 1 copy per 33 EVs.
For NT4, so for NT4 it was right. 6. We found to be enriched 6 around the average of 6 copies every 100 extracellular vesicles.
That's for the IMAX, right?
IMAX. Right. So it was exactly 4 every 100 extracellular vesicles for CDC-EV, both with ultrafiltration method and size exclusion chromatography.
So
that sounds about what other people find.
Yeah.
The second is we've had a lot of really good results with affinity columns to separate. You have the problem of the heterogeneity. We could talk about what you would have on the column, but right. in our work, it turns out that 10% of the total really have the biological activity when one does affinity columns. The other questions I had were down here. Uptake of M1 versus M2 max at the site of injury.
Yeah, that's a very interesting question. I suppose it depends on the phase of the injury. So in the acute phase, the preponderance of macrophages are probably M1, and then long term they're probably even more M2. What we do understand about M2s is that they're probably more phagocytic than the M1s. As you probably know, the M1 and the M2 paradigm is somewhat antiquated, and there's a broader, finer spectrum of macrophage polarization. But the question that you ask is not trivial. That would be an area of future work, and by isolating M2s or maybe even activating them in vitro, we can see the relative uptake of these two macrophage populations. But that's a thoughtful point.
It would be interesting,
Well, you can, with markers, you can identify M1 versus M2 in the tissues.
Right.
Yeah.
Also, these are MSC-derived EVs.
These are cardiosphere-derived EV, cardiosphere-derived cell EVs. So How
do they compare to MSCs?
Yeah, so MSCs
what's their phenotype?
Well, they have they're positive for CD105, they're negative for CD45, so they're non-hematopoietic. They express a number of markers that do share similarities with MSCs, but we know that they're cardiac in origin, and so they're very much different from MSCs, and they likely function differently in the therapeutic context. They're also multipotent, but
in our hands, MSCs go preferentially to M2s in the injured spinal cord.
Oh, interesting.
And they nurture M2s to differentiate, and then the M2s make secondary AVs that affect the vessels.
Well, that's very much aligned with what we're seeing, because what we're seeing is that they also, CDC-EV signal uniquely to macrophages, and they polarize into what we call an MCDC phenotype, which is probably closer to the M2, although admittedly not entirely M2. But the interesting thing is we've shown in other papers that the EVs are uptaken by, say, fibroblasts, and those fibroblasts end up secreting EVs of a different signature that then promote the effect. And that's one very important nuance that differentiates them from, let's say, a small molecule. So even with a minuscule dose, you could see sort of an amplified effect.
Yeah, and what's the target of the fibroblast EVs?
The target of the fibroblast EVs?
Yeah, the secondary EVs, what are they targeting?
Oh, well, we don't know. This is something that we're still investigating. That's that'll be a very interesting thing to consider.
I think we had like this really nice discussion sharing results of the different labs. So thank you, Philip. And I see that Lucio has his hand up and is online already. So please, I will let you to ask your question first and then go with the others.
Hi.
I'm sorry. Who is this? We don't recognize.
Thank you for your presentation. Very, very, very nice. I have just a curiosity. We have shown that the uptake is not always necessary for mediating AV effect. Sometimes they activate a receptor on the surface of cells. So can you comment on this?
Yeah, absolutely. That's another very interesting question. So from my understanding, the engagement of a receptor on the surface and subsequent uptake, and uptake aren't mutually exclusive. So presumably an EV can bind, let's say, or something on the EV can bind a toll-like receptor or a receptor tyrosine kinase system, but it will still be uptaken. The other thing is we aren't entirely sure. So if you think about the sarcolemma of a cell, there's the heparin proteoglycans that latch onto the exosome or the EV, and then the subsequent signaling that happens. And so blocking heparin might block the EV's ability to engage those receptors. Now, for the endocytic activity, again, we're not entirely sure, the magnitude of the effect, but one thing that you'll notice is that only 50% of the uptake was inhibited, which means that there might be some other effect that we're not accounting for that may include the receptor-mediated signaling that you're seeing. So, but your point's well taken, because there are a lot of ligands that decorate the surface of the EVs that might signal. But then again, a lot of signaling does culminate in uptake.
So Eun-Yi Chung, if you unmute yourself, I think that these time points were a bit commented on before, but maybe there's a follow-up question to that. So Eun-Yi Chung, if you're still here, maybe, well, I think you've commented that in your recap, that you'll do it more, right? So maybe we can then just move to the other question. To the Genevieve Bart, are you still here? Well, if not, I can just ask these questions for her. So what about EV cargo? Is it affected by immortalization?
Yeah, so this is, again, another very interesting question. So one of the biggest challenges that we faced with immortalizing CDCs is that, when you immortalize something, undoubtedly it'll change. There will be some sort of modification that happens. And when we tried traditionally immortalizing CDCs, the therapeutic effect was lost. And so it fell back on us to understand, what is it that we have to conserve during the immortalization process that then retains that CDC's ability and the EVs that it secretes to be reparative? And what we understood was the canonical Wnt signaling pathway has to be conserved through immortalization in order for the EVs to be therapeutic. And so when we do sequencing analysis between CDC EVs from primary cells and immortalized CDC EVs, you see a lot of differences. But there are some microRNAs, there are some RNA classes, including this NT4, that remain conserved, and we believe that's what's driving the therapeutic effect. The other thing that's very important is, in order to consider an RNA cargo to be even qualified to be therapeutically relevant, it has to meet some level of abundance for it to be considered as what we call sort of important cargo. Well, many of the times we and others have been sort of chasing after red herrings of these very trace microRNAs that, even though they're implicated in major reparative pathways, are part of a coherent system, but not driving sort of the therapeutic effect. So when you take all these things into consideration, there's only a few that need to be conserved to be therapeutically active. I hope that answers the question.
I've just seen that she's here, but she doesn't have a microphone. So I think she was happy with your explanation. And maybe now I would like to go to Clotilde. She has a longer question, and I think it's going to be more practical if she poses the question directly. Yeah. Oh, great.
Hi.
Hi.
Yeah, thank you. And I'm sorry we are a bit late, so I'm trying to be short, but I liked very much your presentation, your work, and I also appreciated your final limitation slides, which are indeed very, very important, and it's nice to show them. One thing I'm wondering is, when you're using the 100 kilodalton ultrafiltration process, you are co-isolating not only many types of EVs, but also large proteins or large protein aggregates that are together in your prep when you do the therapeutic effect. So even though I got convinced that the NT4 was really in EVs, and when you follow NT4, you really follow EVs, but when you follow the therapeutic effects, you follow the therapeutic effect of this mixture. not only EVs. So maybe like especially when you inhibit with DINASOR or heparin, you could inhibit also uptake of these other components. So I was wondering whether that could explain why at some point you were showing that you still had some heart retention, I think, of the NT4 in DINASOR and heparin, but no therapeutic effect, which would suggest maybe that the therapeutic effect is from the other components and not the EVs. Did you consider this?
Yes. First of all, it's a pleasure and honor getting a question from you, Clotilde, since you're such a leader in the field. Let me go ahead and address the questions that you raised, which are important. So we saw a lack of uptake inhibition in myocardial tissue, and one of the ways that we interpreted this is that, you know, given the uptake by the heart and the lack of a therapeutic effect, we're thinking this might be because you're inhibiting the macrophage response, which I think is driving most of the therapeutic effect. Now, we have done other studies down the line where we actually use size exclusion chromatography and showed the same sort of therapeutic effects in that very pure prep. So I don't necessarily believe that the EVs alone are the only components of the secretome that's therapeutically bioactive. Although I wonder which of the two components, you know, the EVs or the soluble proteins, that have that enduring effect that we see time and again, because these reflect sort of a more fundamental transcriptomically active effect compared to, you know, a protein response that might be rather transient. Now, I don't mean to be dismissive of soluble proteins, but that's sort of the scale of the effect that we've been seeing time and again. And I don't know, you know, what your thoughts are, but this is our line of thinking.
That's I think that would be really important also to check the side by side the effect of the soluble components versus the EV components of your preps.
Absolutely, absolutely. Because if you're thinking from it, yeah. Go ahead.
Yeah, so there's just a technical detail, because I think in your materials and methods, you said it's a 1000 kilodalton filter. At some point you said 100 kilodalton. So which one is it? Because it makes a difference.
Both. Yeah, we use both. For the majority of the study, we used 100 kilodalton filter. But in the study where we had to show sort of the localization of NT4 and the EVs, one of the ways that we use, so we use an RNase protection assay, but one of the other ways that we used is by size exclusion and using 1000 kD filter. So we used 1000 only in the confirmatory studies, but for most of the studies, it was 100 kD.
Right. Yeah, because if I remember well, a long time ago, we used 1000 kD and we realized we were losing a lot of our EVs.
We were losing a lot, exactly. Yeah. Exactly, we were losing a lot. And that's why we used to use 1000 and we switched back to 100. And by the way, 100 kD filter is what a lot of virologists use to isolate their particles. And, you know, from first principles, an EV is just a homemade virus.
No, I fully agree. It's just, when you do sec after 100 kilodalton, you realize that you have lots and lots of proteins that you separate from the EVs by the sec.
Yes, that's right. That's right.
But no, great. Thank you.
Thank you.
So thank you, Clotilde. And the honor for the last question goes to Berta. If she's still here, please unmute yourself. And, okay.
Yes.
Great.
I'm still here. Thank you very much.
Great.
And super interesting study. And thank you very much for the presentation. My question is just only, why did you choose this 20 minutes injection of EB after ischemic injury? Because, I mean, if one wants to translate it to the clinic or from a therapeutical point of view, so maybe it's a bit tight or so, or I don't know. What was your thinking about it or thoughts?
Yeah, so I will take. Thank you so much for the question. Yeah, so it is essentially based on previous study that we perform using at the really beginning different concentration of extracellular vesicles or different protocols. And we saw that 20 minutes after the reperfusion, both in rats or in mice, injecting extracellular vesicles, so we can see the therapeutic effect of these vesicles. So it is essentially based on previous study on these various models. And yeah, if you want to.
Yeah, well, also to be clear, so there's 45 minutes of ischemia followed by 20 minutes of reperfusion. And this is a model that's used quite extensively in sort of cardiac in vivo assays. You know, with the acute MI, which is that we use in other studies, we ligate the LAD permanently, and then we immediately inject the EVs or the drug product. Now, to your point, this model can only measure cardioprotection. It doesn't entirely measure, the sort of the bona fide regenerative effect that we would typically associate, with cardiac regeneration. But in the clinical context, 20 minutes to reperfusion is not entirely distant from reality, unless you are focusing on another aspect of the model.
Okay, because we are working in stroke, ischemic stroke. And yeah, usually, I mean, so the therapeutic window now in stroke, it's not more than 4 hours or 6 hours, and yeah, for the TPA, for example. And yeah, so I was thinking, so if this is something that happens in.
So in our discussion, you brought something to mind. So when we first published the EV paper back in 2014, we had a similar comment from a reviewer, and they said, well, why are you injecting so close to the injury? Why not, wait and see? So what we did was, in another study, we caused the permanent LAD ligation. We waited 3 weeks until the scar became permanent, and then we injected the EVs to try and mimic, let's say, someone who's had a history of MI and and it's really more closely relevant to the way that caduceus was done, and we saw very similar effects, which indicated to us that there was a very sort of poignant reversal of the injury phenotype, whether it's fibrosis or cardiac function or viable tissues.
Okay, good. Thank you very much.
Thank you.
Thank you.
So Ahmed and Alessandra, thank you again for taking your time and sharing your work with us. I think it was, like you've seen, it was very well accepted, caused a lot of, promoted a lot of discussion, and I think that you'll be busy for a while now. I see that you have a lot of plans, and I just hope to see you at some of the next ISEV events. And of course, I would like to also invite all the participants to join again next week with a new study and new discussion. So thank you again, and have a good afternoon or evening or morning, probably also somewhere. So thank you.
Thank you, Metka. Thanks for the opportunity.
Thank you so much.
Thanks, guys.
Bye-bye.
Thanks, everybody.